Right. Hi, everybody, and welcome to today's program, Aping Human Evolution, What the Early Miocene Tells Us About Human Origins with Kieran McNulty. My name is Briana Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray haired woman in a yellow shirt, wearing headphones in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us or you've attended before, we're so glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of your Zoom interface. We're in a webinar format, so we can't see or hear you. As you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen. It looks like two speech bubbles, so that we can sort through as many questions as possible. The Q&A time really flies by. And the Q&A box is also where we will share any relevant links during the program, so you can keep an eye out there. We'll start with a presentation by our speaker, Kieran McNulty, and then I'll join him here to take your questions. During the presentation, I'll also write answers to some of your questions, at least any that I can answer, as will another member of our behind the scenes team, Dr. Ryan McRae. So now I'll go ahead and introduce our speaker. Dr. Kieran McNulty is the department head and professor of ecology, evolution, and behavior at the University of Minnesota. His research seeks to understand how different factors influence the evolution of and variation in anatomical shape among humans and our ape relatives. He studies intrinsic factors that dictate anatomical form, like body size, growth and development, through high resolution 3D digitization and quantitative analyses. He studies extrinsic factors like habitat, geography, and chronology through paleontological fieldwork at early and middle Miocene sites dating between about 23 and 13 million years ago, fossil sites in Kenya. A core tenet of his lab is that strong support for representation, public engagement, stakeholder consultation, and educational outreach are fundamental components of all good research. So now I welcome Kieran to share his presentation.
Thank you, Briana. I'm very excited to be here for the Hot Topics discussion of something maybe a little further afield from what we're used to thinking about in terms of human origins. For those of you in the audience, I'm wearing short cropped hair with fossil remnants of brown hair, but mostly gray, sitting in a very generic looking office, which is where I call my work home. So happy to talk about my work studying fossil apes and the evolution of apes and what it can tell us about human origins. Before I get to that, I actually want to start with my acknowledgments. I think it's important to have enough time to acknowledge all of the people and organizations that have helped me in my work. And I love this quote from Darwin, where he says, "In the long history of humankind and animal kind too, those who have learned to collaborate and improvise most effectively have prevailed." None of this work was done without collaboration and honestly quite a lot of improvisation. I particularly want to thank the people and communities of Western Kenya, and to them I say, Erok Manu Madwang, the National Museums of Kenya and Kenya's National Commission on Science and Technology, who oversee this work and who collaborate with me on these projects. There's so many people who have contributed, and so we'll just say my once and future co-authors on all of these projects, and then the funding organizations that have made this possible, particularly the National Science Foundation and the Leakey Foundation. And for those of you interested, these are links to the various licenses and some documentation of where I get my images and the scientific studies that I'll be citing in my talk. So let me start with this question: why apes? If we're interested in human origins, why do we want to study apes, and not just apes, but apes that don't live around today, apes that were on the planet as far back as 23 million years ago? And when you look at the apes that are left on the planet today, it's actually just a small sample. And so we have gorillas, chimpanzees, and humans forming the African apes. Orangutans and hylobatids are the Asian apes. And then the outgroup of apes, the monkeys, Cercopithecoidea. I think in human origins, we're used to thinking about human evolution in terms of how we compare and how our fossil ancestors compare to things like chimpanzees and gorillas. those that are alive today that are most closely related to us. But if you think about what we're trying to do in human origins, when we're trying to interpret and understand the adaptive evolution of different features, I want to make the case that just looking at our extant relatives is not sufficient. And so think about a critically important evolutionary trait like locomotion. If we map the locomotion onto this group, what we can see is that all of the apes come from some sort of climbing suspensory background. And we can highlight that using these different colors. So both the lesser apes, the gibbons and siamangs, but also the great apes and humans, come from this ancestry where they were climbing and below branch suspending. This is very different from what monkeys do, where they're walking on all four legs on top of branches. And even humans with our interesting and strange locomotion we call bipedalism, if we go back into the fossil record, we can see actually our fossil ancestors in the hominin lineage also had that suspensory features. So the logical interpretation when you think about this ancestry would be that the ancestor of apes and monkeys would have likely been an above branch quadruped. And this is consistent with everything we know about the fossil record. If you then look at the apes, the logical conclusion would be that the ancestor of the living apes would have been a suspensory below branch climbing locomoting organism. And in fact, you can follow that right up the tree to hominins. And at all of these nodes, based on what we see in the living apes, you would assume that the ancestors were all suspensory climbing primates. What happens if you start to map in the fossil apes? So we've got a number of fossil apes with a good fossil record where we know something about their skeleton and therefore their locomotion. And you can see that a lot of these, shown in green, also would have been suspensory primates, right? They were probably climbing and suspending and clambering in ways that are similar, or at least remind us of what extant apes do today. But there's also a bunch of apes that don't do anything like that, that still have remnants or still fully practice a more above branch quadrupedalism. And when you start to look at the ape record nestled within the extant apes, it becomes much harder to justify inferring that these ancestors were suspensory locomotors. And let me just add, it's a little hard to see, there's another 15, 20 different types of ape we don't know yet because we haven't found enough of their fossils to interpret their locomotion. So if we limit ourselves to just the living apes on the planet, we potentially are going to draw conclusions about not just ape evolution, but also human origins that are based on false information. Add to that, layer onto that, we have no fossil record of chimpanzees and gorillas at all. We have a handful of teeth from chimpanzees that are about 500,000 years old, and no clear record of gorillas. And so when you put all of this together, it makes it very difficult to really interpret what these ancestors would have looked like. And from that perspective, you really do need to understand this ape record in order to start to infer the evolutionary origins of the hominin lineage. Another reason to look at the apes is because they provide a different context and a different way of thinking and interpreting the features that we often associate with our lineage. So when we think about what does it mean to be human, we're often thinking about things like large complex brains, upright walking, increased body size, tool use, advanced food procurement, small canines, small teeth, opposable thumbs. Lots of features that we associate with hominins. And we see this in the hominin fossil record, either in incipient stages or fully advanced when we get to modern humans. Now, if you take this list and look at those features that we actually find either fully developed or partially developed or initiated within apes, it's actually quite a bit of the list. We can actually look in apes and find evidence for a lot of these different things that we typically associate with humans. And so that gives us a different system to either ask the question, why did this evolve independently in both, or why did it evolve in the shared common ancestor? If we push it back to the early Miocene, the time period that I'm interested in, we still see a substantial list of features that we associate with human evolution that are found or initiated among the early Miocene apes as far back as 21 million years ago. And so this gives us a vector into understanding and interpreting why humans evolved the way that they did. So if you're interested in the origin and diversification of apes like I am, the place and time to go is the early Miocene of Africa. Africa at this time had been an isolated island, and due to plate tectonics is just about to merge with the Eurasian supercontinent. And this causes a number of really interesting interactions. So if we start here about 35 million years ago, we move forward in time, we can see that really important events in hominoid evolution, the evolution of apes, map on pretty well to important events in the tectonic or geological record. And so as Africa docks with Eurasia, we're going to see the closure of the Tethys Sea. We're going to see rifting in the Eastern African rift system, acceleration of the Tibetan uplift. This all leads to changes in climate and temperature. We're also, as you can imagine, going to start seeing in the fossil record evidence of flora and fauna moving between Africa and Eurasia in ways that they couldn't before, right? Once that land bridge is connected, then we have flora and fauna migrating much more freely. And this is going to set up another series of selective pressures. So there's a lot of really interesting things going on in the early Miocene, and this is the time when we see the first massive diversification of apes that seem to split up into lots of different species, most of which seem to go extinct, but some of which persist for millions and millions of years. So I want to focus primarily on Kenya, and this is where I've done the majority of my field work. I'm going to talk a little bit about work I've done collaboratively with my colleagues in Uganda, but for the most part, I want to focus on Kenya. And you can see from this topographic map, in the western third of the country is the Great Rift. And what we often don't pay attention to is jutting westward off of that rift is a smaller rift, the failed rift arm, called the Nyanza Rift. And if we zoom into the Nyanza Rift, we can see that at either end of the rift are volcanic edifices. One is the Kisangiri volcano, the other is the Tinderet volcano. And all of the early Miocene sites that I work at are associated with these volcanic edifices. This is where the vast majority of early hominoid fossils comes from, these sites in the Nyanza Rift. There's also middle Miocene sites that my student Samuel Muteti is working on. And so together, these fossil sites allow us to really think about a transition from the early Miocene and some of the oldest fossils in the Miocene, transitioning into the middle Miocene, where we start to see not just apes, but actually apes that are probably more closely related to living apes. So it's a great window of time to look at the evolution of apes. So I want to talk about three hypotheses that are that we can look at in the early ape record that have some bearing on how we interpret human evolution. And the first hypothesis is that key ape features, things like being orthograde, things like having more versatile locomotion, key ape features first evolved in dense forests. The second hypothesis is that locomotor versatility in early apes evolved specifically to promote terminal branch frugivory, specifically to allow apes to climb to the ends of branches despite their large body size and access fruits. And then the third hypothesis is dealing with a specific ape called Akembo, and Akembo is the best preserved, best represented fossil ape in the record. And there's some argument as to whether it's actually an ape or not. So I want to talk a little bit about how we can test whether it's an ape. So to think about these hypotheses, first I want to tell you a story. And it's a beautiful story. It's a story we've been telling ourselves for hundreds of years. And it goes something like this. Back in the early Miocene, 20 to 23 million years ago, Africa was covered with a Pan-African forest, a forest that stretched from one coast to the next. And as time went on and the earth cooled and dried, that forest started to shrink. As the forest shrank and the grasslands got bigger, our ape ancestors had to come out of the trees and they started walking upright and they became bipedal and they eventually evolved into humans. This is a story, parts of which actually go back to Lamarck and Darwin, who talked about the importance of grasslands in the formation of our lineage. But it really got its scientific traction with Raymond Dart. And he talked about what we now call the savanna hypothesis. And I won't read this entire quote, but he argued that in order for humans to become something different than their ape ancestors, they needed to sharpen their wits and quicken their manifestations of intellect. And this had to happen in a more open, veldt-like country, speaking from South Africa. And so Dart really solidified this idea that it was moving out onto the grasslands that helped our ancestors become what we are today. So I'm going to challenge that a little bit. And I want to think about not specifically the human evolution part of that, but this early Miocene part, where Africa was purportedly covered in a Pan-African forest. And so if we're going to think about that from a scientific perspective, we need to ask, what kind of evidence do we actually have to test that? And so in the fossil record, we have things like plant macrofossils, where we see leaves and grasses that preserve. We actually see casts of trees. This is an example of a tree trunk with roots that we found had about 500 fossils inside. We have things like phytoliths, and phytoliths are silica bodies produced by plants, and because they're silica, they preserve for a very long time. And they also have fairly good taxonomic value, that we can match certain types of phytoliths to certain groups of plants. So they're really good at reconstructing vegetation. And finally, we often look at stable carbon isotopes and the proportions of different carbon isotopes that we find in things like petagenic carbonates or in tooth enamel can help us to make determinations about whether these were very closed environments or more open environments. So this is the kind of evidence that we can look at to test this hypothesis about a Pan-African forest. And there is some evidence. This wasn't something I set it up as a cute bedtime story, but there of course was good scientific evidence for this. If you look in general at the evolution of grasslands in low latitude environments, we don't see global dominance of grasslands until about 10 million years ago. And so if you look across these different land masses here on the right, it's really at about 10 to 8 million years ago when grasslands at low latitudes really start to take off. If we look in the African record specifically, and these are different ways to look at carbon isotopes, we see the same thing. In Africa, it's about 10 to 8 million years ago that we see this record of more open habitats. And without getting into the details of isotope analysis, what you're looking at in this chart on the left is that prior to 10 million years ago, these isotope proportions are way over on the left of the graph. When you get to about 10 million years, they start to veer off to the right side of the graph. And that's indicative of more open habitats. But there have been hints that this story is not that simple, that there's minimally some nuance to this. And this is a paper published by John Kingston. This graph is from a paper by John Kingston published in 1999, where he looked at a site called Fort Turner, which we date to about 14 million years old. And I've highlighted the samples from Fort Turner that show actually it looks like there may have been some C4 grasses, or at least C4 vegetation, as far back as 14 million years ago. When you combine that with some of the other ecological proxies that we have been getting independently, it suggests a need to look at this question much more carefully. And so in thinking about how to do that, thinking about how we could possibly test this as far back as 20 to 21 million years ago, Some colleagues and I came up with a project that we called the REACH project, and REACH stands for Research on the Eastern African Catarrhine and Hominoid Evolution. And it was something of a unique project in paleoanthropology in that we actually got together and collaborated with people who work at all of the early Miocene fossil ape sites in Eastern Africa. The vast majority of us had never worked together before, didn't really know each other, but the value of the science or the potential value of this kind of collaboration was sufficient that we all pulled together and started working in ways that are very rare in paleoanthropology, I would say. And the goals of the project were, again, to sort of take a regional perspective rather than having all of these individual isolated projects, to share data collection protocols, to use the same specialists, and so we could compare results, to conduct multidisciplinary analyses with overlapping sampling so that we could validate and corroborate each other's results. And then most importantly, to also integrate our local research partners and collaborators in ways that were not just separately site by site, but actually in a broad research collaborative network. This is a photo of one of the workshops that we had and some of the participants that were able to attend. And this project funded 6 years of fieldwork and resulted in tens of thousands of fossils recovered and what we think are some fairly important advances in our understanding of ape evolution. So let me show you one of those advances specifically as it relates to this hypothesis about the early Pan-African forest. So I'm going to show you again, this is a graph from one of our papers, and this is basically replicating the data from the literature that you've already seen. And so again, whether you look at the pedogenic carbonates on the far left panel, soil organic matter on the next panel, leaf wax isotopes, or phytoliths. So those are the 4 panels, and then the 5th is looking at atmospheric CO2. You see the same sort of pattern. Back in the early Miocene, our samples are skewed over to the left. When we get to about 10 million years, things start to either skew to the right or broaden to include both the right and left side of the graph. Again, indicating more open habitats after about 10 million years. What happens when you map on our new data, most of these sites had never been fully sampled for this sort of analysis. When you map on our data, what you find is that the early Miocene isotope record actually spans almost the entire range of variation that we see in the Pliopleistocene record. In other words, back in the early Miocene, we had not just closed forests, but we had grassy forests and woodlands. We had wooded grasslands. We had fairly open environments and very closed environments, not just a single Pan-African forest. The phytolith record also confirms that. I'm not going to try to deconstruct this entire graph, but essentially, if you look at where these fossil phytolith assemblages place us at sites like Moroto, Bukwa, and Rusinga, what you find is that there was as much as 60% potential C4 grasses at some of our sample sites. So these were not closed forests all the time. Some of them weren't even woodlands, but much more open environments. A multidimensional analysis confirms that all of our fossil sites, again, more similar to wooded grasslands and bushlands rather than to the forests you see over here on the left of this graph. And if you're interested, these are what phytoliths look like. These are examples of phytoliths that are specifically associated with a clade of C4 grasses. So pretty good evidence that we had not just open areas, but C4 vegetation. So when we think about our hypothesis that key ape features first evolved in dense forests, what we find in the early Miocene with the first real adaptive evolution of apes, what we find is a variety of habitats, both dense forests, but also very open wooded grasslands. And so for my colleagues and I, this is a good reason to reject that hypothesis. Not that there weren't dense forests, but that we can't immediately conclude that any of these 8 features evolved in that kind of habitat. So moving on to the second hypothesis, that locomotor versatility in apes evolved to support terminal branch frugivory. I want to look specifically at one of the sites in the REACH project called Moroto. And so we were down here in the lower left of this map in the Nyanza Rift. We're going to go straight north to the top of this map to Moroto. And this is one of the oldest early Miocene ape sites at 21 million years old. And it's known for Morodopithecus, which has some good craniodental material and some very fascinating postcranial material. And one of the great things about Moroto is that all of our ecological proxies, we were able to sample very well from a fossil layer that is also the best layer for finding fossil apes. And that's this layer here. And so we were able to specifically put these ape fossils within the context of a highly resolved paleoecology. And that's something that's very rare in the fossil record. What that habitat looks like is much more open than we would expect. No evidence for a dense forest here, but probably more of a woodland or even a wooded grassland. And so these fossil apes specifically, we can place in that more open habitat. But what was interesting is if you look at the dentition of Morodopithecus, and so you see Morodopithecus here is the tooth on the bottom, and that's compared to two species of proconsul just above it. And if you just look at the tooth, you can see the proportions of the tooth are quite different. And those proportions map on fairly well to differences between frugivory and folivory, where frugivores tend to be much higher on this graph here from McClatchy et al. And that represents their much broader, more squarish appearance. Morodopithecus and other folivores have a much more narrow rectangular tooth. And so this suggests that Morodopithecus was, at least in terms of its adaptations, not adapted to frugivory at all, but much better adapted to folivory. So if we look at this composite picture of the best sampled paleoecological site at Moroto, it suggests that Morodopithecus was inhabiting a grassy woodland adapted to eating leaves rather than a dense forest eating fruit. So again, for my colleagues and me, this would be reason to reject this hypothesis that locomotor versatility evolved specifically for terminal branch frugivory. So my third hypothesis then is that this particular ape called Ikembo was in fact an ape at all. And there's been a lot of argument over whether it was an ape or something that that preceded or whose lineage preceded the split between apes and monkeys. And just to show you graphically what that argument looked like, this is a photo from a workshop put on by the Turkana Basin Institute on ape evolution. And this is kind of the consensus of what all of the participants could agree on. And I will thank Carol Ward for that photo. We can maybe draw a slightly better consensus just to simplify things. And so this is based on a phylogeny from Nengo et al. In 2017. And the question then is, does Ikembo, which I've drawn here, sitting between monkeys and apes, so on the ape side of this clade, does Ikembo truly belong with the apes, or as some have suggested, does its phylogenetic position actually come down preceding the split between monkeys and apes? And so this is still an open debate. And testing this is could be done in a variety of ways, but my favorite way to test hypotheses in the fossil record is just to find new fossils. And so let me show you a few of the fossils we've found in my years working on Rusinga Island. And you can see we found some pretty decent material. A couple fossil facial skeletons, palates, jaws, arm bones. And to think about this particular hypothesis, I want to focus on this one specimen. And this is a facial skeleton found by Cliffo Chiang in 2016. And this particular specimen is critically important for one reason, and that is it's not warped. The vast majority of fossils on Rusinga Island are warped, and we can try to unwarp them using our brains or using math, but that requires making a lot of assumptions. This specimen is actually unwarped. It broke instead of warping, and that allows us to apply methods without making those assumptions where we can more rigorously and statistically test hypotheses. And so I used what we call the standard geometric morphometric toolkit based on Procrustes analysis. So putting anatomical landmarks on the specimen, like you can see in this skull up in the upper right, creating mesh representations, and we can use those 3D models then to do some fairly high-powered statistics. And so one thing that we've done is we've looked at phylogenetic comparative methods, where we've taken those anatomical landmarks and those wire meshes, not just from our fossil, but from all of the specimens you see here. And so we sampled a third of all of the living monkeys, more than half of the living ape species, and the half that we're missing are just gibbons. I shouldn't say just gibbons, but their crania all tend to look very similar. And then we threw in some platyrrhines as well for good measure. And the goal of these phylogenetic comparative methods is, in part, is that we can actually reconstruct what the ancestral state might have looked like based on these living specimens and based on our knowledge of how they evolved, both in terms of the topography, but also the branch lengths in this phylogeny. So we're going to statistically estimate what these different nodes look like and try to better understand where our fossil ape Ikembo sits. Just to show you what something like that might have looked like, this is an example of the estimated ancestral catarrhine. So this would be our estimate of the ancestor of monkeys and apes. So this skull does not exist. It's not a real object. It's a statistical estimate based on phylogenetic comparative methods that shows us what that ancestor might have looked like. So here's the actual analysis, and this is an incredible mess, and I'm not going to ask anyone to try to understand it, but let me just show you a couple highlights that are relevant to our hypothesis. So these are all of those monkeys and apes that we sampled. All of the colored dots represent extant specimens, and all of the white circles represent those estimated ancestral forms. And so those are the gibbons and siamangs, those are the great apes. Here's our estimated ancestor of monkeys and apes, our estimated ancestor of the living apes, our estimated ancestor of the great apes. And so the question is, where does Ikembo sit within this plot? And surprisingly, at least surprising to me, it not only sits among the apes, but it sits among the great apes. Now, I'm not arguing that this was a great ape. That argument has been made. There's some merit to it. But if nothing else, I think this is a good reason to say we cannot reject the hypothesis that Ikembo was truly an ape, certainly as far as its cranial skeleton goes. So key takeaways from my talk, things that I hope you got from the talk. The human lineage emerged from fossil apes. This diverse and speciose group provides a crucial framework for interpreting human evolution, and that's a framework we cannot just infer from the handful of living apes that did not go extinct. There are several key features in hominins that first emerge among the early Miocene apes. So the early Miocene apes thereby both anchor our phylogenetic tree, but they also provide corroborating or additional context in which we can look at how these kinds of features evolve. I think it's also useful to get this perspective because it reminds us that adaptive evolution is really a multifactorial process. If we think about something like the evolution of bipedalism, the question I often get in class is, well, what was the selective pressure that made us become bipedal? But when we think about a process that was going on for 5 million years, maybe we shouldn't be thinking about a selective pressure, but a whole multifactorial process through which this adaptive evolution occurred. The early Miocene in Eastern Africa is one of only a few places where we have this level of diversity of catarrhine primates, where we have this long a geological record, and we have a wide variety of habitats across which at least some of these apes persisted. And perhaps the most important point, the key results from that I presented today could never have been obtained from a single fossil site. Not one of those 20 different fossil sites in the REACH project had sufficient evidence to say what I've said today. But together, the evidence is actually overwhelming. And so this kind of collaboration has a lot of power to answer questions that we often think we can't ever get to. Looking more specifically at our hypotheses, I would argue there was not a single Pan-African forest in the early Miocene. There was likely multiple cycles of forestation and deforestation throughout the Cenozoic. But I do want to point out, for those of you interested in human evolution, I'm not saying the savanna hypothesis is dead, that there was a rapid aridification and expansion of the grasslands in the Pleistocene, and that certainly has a lot of explanatory power in other aspects of human evolution. But my early apes were living in a variety of habitats, including fairly open ones that included C4 grasses. And so we don't see any support for this traditional model that early apes evolved in a dense forest, mostly to eat fruit. And finally, the best known of our early primates, Akembo, has features that are consistent with the clade of apes and humans. And so I would argue that it is, in fact, an ape. So I'm running a little long, but let me just end with something related to our work. And that's the idea that we can actually use paleontology not just to advance science, but to improve the lives of the people in whose communities we work. And so this is something that we started all the way back in 2006 when we started doing our field work, was not just thinking about the kind of science we want to do, but how does that science also benefit the communities and the regions that we're working in. And so we've taken a multi-scale approach to this, engagement and outreach across lots of different scales, from training scientists to supporting and developing institutions, to doing local outreach, training educators and local collaborators, in a sense, really just becoming part of the community. And we hear a lot about this these days in the past several years. I can tell you that this is a good idea, not just because it's the right thing to do, but in fact, it actually has tangible scientific results. Just to show you a few of the things that we've done, this is a medical clinic that we started working with the local community to build in 2006. One of my favorite activities, this was a bridge to get to one of the most important fossil sites. If you can imagine trying to carry equipment across that, it was not at all fun. It was also not fun for the community, who had to navigate this bridge to get their goods to and from market. And so we were able to work with them to build a more substantive structure. And then my team started almost 10 years ago now, supporting a local school for orphan and vulnerable children. It's a scary thing to do. We don't know where the funds come from. We don't know how we're going to support people from year to year, but we do it, and everyone benefits from it. And everyone benefits because once you're part of the community, not only do you find more fossils, but you find better fossils. When you've got 40,000 people who are on your team helping you do your work is going to be better. It helps us better implement our research agenda, because our research then becomes part of the community agenda. It means that when we have trouble, we can find local solutions to our local problems. It means long-term community investment helps to maintain and protect our interests. And it means people we're working with will better trust our advice and services and the infrastructure we provide. As I said, the reason to do this is because it's the right thing to do, but here are some tangible effects that actually just make the science better. In the end, it provides great job satisfaction, and I can tell you we can talk about how we measure success and the metrics of whether this is good or not. This is my favorite measure of success, when one of my team members pulled up in a motorbike that he had branded Akembo after the local fossil ape. So with that, I will leave you with a picture from field camp on Rusinga Island. No, it is not a horrible, difficult place to work and live. It is a beautiful place on the shores of Lake Victoria, and I encourage all of you to visit. Thank you.
That was wonderful, Kieran. Thank you. And I really enjoyed you talking about your efforts with the local community, particularly at the end. We have questions coming in already. Our behind the scenes teams has also has a series of questions. So I'm going to start with one of the audience questions from Niels, who asked, is specialization to one kind of locomotion or the other, like above or below branch, is that a one-way evolutionary trip, or could a lineage or species go back and forth between the two?
That's a great question. I have never been a proponent of Dollo's law, this idea that evolution only goes in one direction and doesn't go backwards. So I think it very much can go, back and forth. And again, when we think about the millions of years we're talking about, then you have to say almost, of course, it can go back and forth. One of the interesting things about ape evolution is it's actually becoming more versatile rather than more specialized in some ways. And so that, might give apes a variety of different ways that they could then evolve. And that may in part explain why it's been so challenging to sort of think about not just the direction of ape locomotion evolution, but also the origin of humans.
Yeah, that makes sense. I have a smaller sort of specific and maybe technical question. I think you described that a lot of the fossils on, I think it was Rusinga Island, are often warped. Can you sort of talk about that? Like, how does that happen? Yeah.
Yeah, I can, and I'm not a geologist, so I'm just repeating what John Van Coovering told me. John Van Coovering did did the stratigraphy on Rusinga Island for his dissertation as part of Louis Leakey's project. And we took John out to Rusinga in our very first field season, so he could orient us to the geology. And I asked him this question, and I'm just repeating what he told me. It sounds crazy. What John told me is that the sediment on Rusinga is so densely packed that when it shifts, the fossils don't have enough space to break.
Hmm.
That they literally can't break because there's no space to do it. And so they warp instead. Now, that doesn't mean they don't break, right? So you do get these longitudinal cracks, but I would say 90% of the fossils we find are warped in some way if they're
Huh.
more than just a tooth.
Interesting. So here's a comment and a question from Renee, who says, great talk. Are the ape associations with paleo environments based on presence absence, or is there a measure of abundance?
Great question. I have a PhD student working on this, not just for the apes, but also for the mammals. Um, so I could do a whole different talk on Rasinga, which is a wonderful and fascinating place. But we previously thought that all of the 360 so meters of deposits on Rasinga, most of which are fossiliferous, we previously thought all of that accumulated in a really rapid time. And so no one had bothered to look at, things like abundances and proportions. Thinking about paleoecology and mammal communities, it was kind of a checklist. It's like, oh, yep, we've got that species, we've got that species. And so look, everything's the same. Now with new dates, we know that the sequence goes for about 3 million years. And so my PhD student is tackling this question. What does it mean that maybe the species are the same, but the proportions are changing quite a bit? And a really simple example, sorry, this isn't about apes. Really simple example is the rodents Diamantomes versus Paraphiomes. And in the oldest localities, you get almost entirely Diamantomes and almost no Paraphiomes. And as we get to the younger localities, we get just the opposite. The challenge to doing this work, so sorry, the question was about apes. The challenge for the apes is, even for the apes, we don't always have great provenience. And so we don't always know exactly where they're from, or if we know which site they're from, we don't necessarily know which layers they're from. And that the other challenge with the apes is they're rare. We have a ton of them because we have a ton of fossils, but it's not like Songhor, where the primates are very abundant, or Capertay, where that bridge was. Capertay, the primates comprise more than 40% of the fauna, which I've never seen anywhere else.
Mm-hmm.
So on Rusinga, where we have a bigger sample of apes generally, or a better preserved, it's only because we have, tens of thousands of fossils.
Hmm. And I think because the question was the ape association with the paleo environment. Um, so I think it may have been a mammal community question actually about like
Yeah,
presence, absence versus abundance.
I will say, this is a pitch for funding, of course. Um, when I have tried to talk about the value in trying to understand primate adaptations by looking at places where we don't have primates or have very few primates, I have not gotten any traction. But to me, a big part of the question is, why don't we have apes at sites like R4 on Rasinga, at sites like Kurungu, where we got thousands of fossils
Hmm.
and just a couple apes. So I think there's a ton of work to be done toward answering that question.
Excellent. Here's a question from, looks like Gerald Alpern. Does genetic research on existing primate lineages reveal anything about the timing of branching points among monkeys, apes, and hominins?
Yes. So the genetics provides a really useful scaffolding for thinking about these timings. What a lot of people don't know is that scaffolding is often calibrated by the fossils. So there was a massive study done on primates, came out, I want to say 2 years ago, sampled hundreds of modern primate species, and it had all of these divergence times, which are very useful. But then if you dig into the fossils used to calibrate it, then maybe you start to question a few of them. This is what we do, of course. So yes, it does provide a really useful framework for starting to think about it. What I would love to see is much more close collaboration between the geneticists and the fossil researchers, where we're all kind of doing the same thing from our different perspectives. So I'm looking at the genetics and trying to understand my stuff without really working with the geneticists, and they're doing the same. And if we work together, we could actually come up with probably really robust models of when these divergence times are.
Hmm. Sounds like a good future collaboration ideas. Here's one of our behind the scenes questions. Could you talk more about the origin of the REACH project? How difficult was it to get different researchers, this big group to work together? And what's the future of the REACH project?
Yeah, great question. The origin of the reach, I mean, it wasn't quite, but it was almost literally the slide I showed, with some weird ape sitting up in a tree looking at the volcano. I was the weird ape. I wasn't a tree. I was up on a cliff. Um, and it was when I got my first big grant, the first field season after getting a big grant. I'm like, oh wow, I've got all this money and all these great smart people surrounding me, and like, wow, I can't answer the questions I want to answer because I'm stuck at this volcano. And why in
Mm,
the world would I think that my volcano is going to provide, the answers to all of the early Miocene? I mean, it makes no sense. It's a very myopic point of view. So then I guess I started thinking, well, what do we need to do to answer these questions? What kind of data will we need? And um, the paper by Richard and Meve Leakey, the Afropithecus and Turconopithecus papers, actually one of them has a little blurb at the end that sort of talks about, hey, we need to start thinking more regionally. And from there, the way I started putting the project together was I started inviting people to the University of Minnesota to give talks and then to kind of pitch this idea. And again, these were people I had not collaborated with and people I didn't know. In
mhm.
some cases, people I really looked up to. And it took some convincing because everyone's got their own sites and their own agenda, and I think the reason the project worked as well as it did, and there were many difficulties, but I think it was because the goal was not to supersede anybody's scientific agenda. It was to bring them all together and
Mhm.
weave from those separate agendas a bigger picture.
Yeah, excellent. I think, these big collaborative projects are incredibly useful.
Sorry, to the future
Oh,
of
yeah.
the project. So you know, the funding ran out, of course, but the goal of the project, okay, it was the funding, but also was to build these collaborations, and the
Hmm.
collaborations are still there, and they have moved on in ways that don't just map onto the original groups. And so a lot of researchers now are sort of working together in different ways and at different sites, and we are talking about, you know, sort of refocusing and reorganizing, and what do we want to do next. So time will tell.
That sounds exciting. So this question follows on that one nicely. This is from Alec, who says, "Thank you so much. How often do you go back to the site to collect more fossils, and do the school students in Kenya ever get involved?"
Great question. I go to Kenya twice a year, so I go between semesters in January, and primarily to work in the museum because everything you find in Kenya stays in Kenya. But I do go out to the field a little bit, mostly to Rusinga to work with the school, actually. But you know, you get out to the sites and collect a few things. In the summer is when I do my field work, and so I will be out at the fossil sites anywhere from 2 to 6 weeks, usually depending
Mhm,
on funding and the research aims.
mhm,
The kids at the school that I support do not help in the fossil collection. So the school is 3 to 10-year-olds, so they're
mhm,
a little young to be helping out. We have been working with some secondary students and some university students on Rusinga specifically to, you know, not just get their assistance, but also, you know, train those who are interested, with the possibility of going, you know, going on and maybe even getting a PhD someday.
Excellent. Thank you. Here's a question from Marianne who asks, "Why didn't you include bonobos with the great apes?"
So I'm not sure which place I did not, but in my head I did. And
Fair.
part of the issue with me, it's not an issue. It's both a simplification, but it's also the idea that usually, commonly, with a lot of exceptions, but commonly, um, the genus corresponds to the common name. And so because bonobos and chimpanzees both are in the genus Pan,
Hmm.
I usually refer to them as chimpanzees. Um, but I, you know, certainly recognize a separate species. But by the same token, I recognize 2 species of gorilla and 2 or 3 species of orangutan. And so, in essence, I oversimplified the whole mess.
No, I do that too. I often say just chimpanzees for chimps and bonobos. So here's a question from Nick who asks, "Is there a substantial difference in the usefulness or ease of obtaining paleoecological data, ecomorphology, hypsodonty, mesowear, from associated fossil fauna at early Miocene sites prior to versus following the beginning of faunal exchange with Eurasia about 19 million years ago?" And then he says, basically, are the preferred methods for determining these paleo environments different depending on the date of the site, or is it just case by case on what you can recover?
So you always have to start with the latter, right?
What's there?
What can you actually recover? And
Hmm.
that, I mean, that radically determines what you're able to do.
Hmm.
A great example would be a site that used to be called Mesva Bridge, now called Tonde Bridge, and it's a chock full of fossils. All of those fossils jammed into a space that was maybe 2 meters by 2 meters by 2 meters. I mean, it's a tiny little cube of fossils, and it's also the oldest published Miocene ape site. So that would be a great place to sort of look at what are things like at 21 million years ago versus, you know, these different times, and we actually have good evidence of multiple sort of waves of faunal exchange.
Mm-hmm.
And that's a really interesting line of work that my colleague Susie Cody works on. Um, but so at this site, Tonde Bridge, like we would love to be able to do this kind of really in-depth mammal community analysis and ecomorphology, but there's
Hmm.
just not enough material. It's like the best it's at just the right time that we want to sample, and just not enough fossils. So yeah, absolutely have to start with, what can we possibly do? Um,
Mm-hmm.
and then work out from there. And what we found in the REACH project, and honestly, if you read any paleoecology paper, they all kind of end up saying the same thing, which is the more proxies you have, the better. Right?
Mm-hmm.
Every proxy you have is going to be flawed in some way. And that's one of the great things about the REACH project was not only do we have multiple proxies, but we were using the same proxies at multiple sites. And so when you have, excuse me, when you have very faint evidence of something again and again and again and again, then that starts to be more meaningful than, well, okay, maybe this was sampling error or something.
Mm-hmm.
Um,
Oh, go ahead.
oh, I was say, so yeah, you start with what you have, and then
Mm-hmm.
ideally work it into multi-proxy analysis. Most of those proxies are much more dependent on soils and soil types and habitat and habitat types rather
Mm-hmm.
than the faunal exchange. But yeah, if you start to get into the ecomorphology, what does it mean, for example, what does it mean to look at the astragalus of a tragulid, which is what we have in the early Miocene, versus a bovid, which is, I don't want to say replaces them, but
Mm-hmm.
lots of bovids in the middle Miocene, no bovids in the early Miocene. Can we interpret the ecomorphology of their bones the same way? That's not clear. And so, sorry, this is getting
Mm-hmm.
long, but I guess my final point in that question would be, what we do a bad job of in paleoanthropology is thinking about not just what's the best answer we can get, but how good do we think the answer actually is? And
Mm-hmm.
providing kind of error bars for, hey, here's what the data tell me, and I can't get the data to tell me anything else, but here are all the reasons why it might be something else anyway.
Yeah. Mm-hmm. No, I agree. And actually, I'm going to answer. We have time for two more questions. One, which I'm going to insert, is one of our behind the scenes questions, which I think is relevant here. And it's, if different paleoclimate proxies disagree, how do you rectify that? Or are certain ones generally more reliable than others?
Yeah,
I know, big question.
I don't have an answer to the first one
Yeah.
because it really does seem to be specific to each case. And
Yeah.
so, I mean, our
That
three
makes sense.
thing is a great example. We have actual tree trunks that we map on the landscape, and we can use that mapping to predict what the forest looked like. And it was a
Mm-hmm.
dense, multi-canopy, multi-story forest. But we also have paleoecological proxies that suggest there were some open spaces.
Mm-hmm.
And so how do you reconcile those? Well, there are a few different ways. So you definitely want to interrogate your proxies. How good are these
Mm-hmm.
proxies? Do we expect them to fail for some reason, for some sampling reason, for some geochemical reason? And then you also want to think about, well, why would we imagine that there's some sort of pure landscape that's only
Right.
this thing, right? And so you
Mm-hmm.
have Cavalli Forest, which is a forest with these lovely open spaces.
Yeah.
And so, in essence, it's not a great way to do science because you're kind of back engineering how to interpret your data.
Mm-hmm.
But sometimes that's the best we can do, and that's
Yeah.
again why I say it's important to put an estimate of how good your answers really are in the first place.
Yeah. Agreed. All right, we have time for one more question. And this question is from Tad, who said, I'm interested in the origins of war among humans. I understand that these are pre-humans, but is there any evidence that these ancestors 20 million years ago fought with each other, not as individuals, but in groups?
The short answer is no. We do
Hmm.
not have evidence of that. Um, but when I try to imagine what that evidence would look like, it's hard to imagine how we would even find it if it did exist.
Hmm.
Um, you know, without having a kind of messel like, you know, they literally preserved while they were
Right.
doing whatever. So no, in that sense, we don't have evidence. We can look at modern monkeys and apes and sort of imagine the kinds of interactions they had, which include
Hmm.
certainly living apes, things that are very, very warlike.
Hmm.
And part of what, again, my students and I are doing is trying to get a sense of how well can we say that, for example, there were two species of akembo at the same time. Like
Hmm.
we have them in the same layer at the same site. Do we have that kind of resolution? And that's a question we're still working on. And when we get there, then we can start to think, I think, more critically about how they might have interacted.
Yeah, excellent. What we can and can't answer, always big questions. Okay, so this concludes today's virtual program. So please join me in thanking Kieran for sharing his work with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Dr. Ryan McGray, who works with us here in the Human Origins program, to our donors, volunteers, and viewers like you, and finally, to all our partners who help us reach, educate, and empower people, millions of people around the world today and every day. Thank you. We'll be back next month on Thursday, December 19th, with Dr. Shara Bailey from New York University, who will talk with us about the extinction and legacy of the Neanderthals. We've put a link in the Q&A where you can find more information about our upcoming programs and how to sign up for the museum's weekly e-newsletter. That's the best way to stay informed on upcoming programs and learn more about the museum's research and exhibitions. After this webinar ends, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future Hot Topic programs, and we appreciate your input. Again, thank you to our participants, thank you, Dr. McNulty, and thank you to you, the audience. We'll see you next month.